All right, so now we're going to start talking about each of the 8 indicators and how we measure them. So we're going to start with the biological indicators, and we're probably going to go into the most depth about the aquatic invertebrates because it's a bit more complicated, but it's a particularly important indicator for this method. We might take a break after we get through the aquatic invertebrates before going on to the subsequent ones. Okay, so the aquatic invertebrates, we have 4 metrics used in the Western Mountains SDAM to characterize aquatic invertebrates. There's the total abundance of aquatic invertebrates, the total abundance of mayflies, the total abundance of perennial indicator families, and the total number or richness of perennial indicator families. We have these 4 different metrics. Some of them are used in the snow-influenced regions, others are used in the non-snow influenced regions, and some are used in both. So it kind of varies, but these are all things you can get out of your data pretty easily. The way we collect aquatic invertebrates is identical to what we do in the arid West. We do a 15-minute search that covers at least 6 distinct locations within the reach. After you've collected them, you need to identify them to family, and the time you spend sorting and IDing doesn't count towards those 15 minutes. I want to stress that stream-side field identifications are absolutely appropriate and acceptable, but for some people it might be better or easier or more cost-effective to do lab identifications. So both of these are acceptable for this method. The invertebrates that you count, you don't distinguish between living or non-living organisms. So whether it's a whole larvae versus just a cast skin, or the case of a caddisfly or a shell of a snail, those all count the same as an individual organism. We want to make sure to target all the habitat types that are available within the reach. Typically, areas like riffles are among the most rich and will have the greatest diversity of organisms that you collect. But if you have areas that are like pools or undercut banks, you want to make sure to sample them as well to get you the full breadth of organisms that are likely living in that reach. Each of these different habitat types have appropriate methods for sampling them, and we'll talk a little bit about each of them. So in locations with like fast flowing water, like your riffles and your runs, you definitely want to start downstream and work your way upstream. A D-frame net with like canvas sides, these are generally the most effective way to collect these organisms. You put the D-frame net in the water perpendicular to the direction of flow. You want to make sure the bottom is flush with the stream bed and that net is fully extended and there's no obstruction disturbing the flow. And then you want to spend, maybe 30 seconds to a minute disrupting the square foot of stream bed upstream of the opening of that net. You then want to take the contents of that net, empty it into a sorting tray, so you can go out and collect more without risking loss of any organisms that are already caught in your net. If you're in a location with still water, you can't rely on the flow to actually push the bugs that you dislodge into the net. So basically what you would do is you put the net, again, in the water and you disturb the substrate in front of the net, but then you rapidly sweep the net through the suspended sediment, where all the organisms are hanging out, and you collect them that way. In something like a woody jam, which can be pretty common in certain parts of the Western Mountains, as well as other areas like root mats or undercut banks, you basically are trying to jab them with your D-frame net. And that can help dislodge the organisms into the net. Oftentimes, it's easy enough to just pick up a rock and look at it and see the bugs crawling on it. In this example, it's actually probably not that easy to see, but there is some eggs of a caddisfly, that little jelly-like blob on the top left corner of that cobble. A lot of the clinging organisms will be pretty obvious, like the larger mayflies and stoneflies. You'll see them just clinging to the rocks. And so you can get quite a lot of organisms just looking at the rocks you pick up. sometimes you don't even need to pick them up. You can just see through the stream, and you'll see many organisms without much effort. In fast-flowing shallow waters, where you'll find a lot of things like black flies or net-winged midges clustering, depending on the velocity of the water. If you see these, you still need to do the 15-minute search, because this isn't going to give you the full breadth of organisms living in the reach, but this can give you a quick indication whether or not there's going to be abundant organisms. If you're in a partly dry stream like this one, it's okay to spend the majority of your time in the remaining wetted habitats. That's where you're going to find more organisms than in the dry areas, but you shouldn't overlook the dry areas altogether. You should conduct your search there. In a dry reach, you want to look for the areas where the water likely persisted the longest, and that's where you should probably turn over the cobbles and boulders and substrate and see who's clinging underneath. You can find a lot of like caddis cases attached. You may find the burrow of some organisms, like hellgrammites often create these burrows under cobbles in moist substrate. If you look at the tops of boulders or vegetation around the stream, you might find some skins of larvae like dragonflies or stoneflies. And also you might just find, snails kind of hibernating over the dry period in these sort of areas with remaining moisture. Like I said, you can find living organisms in a dry stream, living aquatic organisms. In the West, there's a genus of hellgrammite called Neohermes, which is actually a specialist that can live for several years in dry streams by building these chambers that help them retain moisture. When flow resumes, they're usually the first ones in the water out there collecting anybody who is unfortunate enough to be caught up in the flow. You should ignore terrestrial life stages, these aerial adults of dragonflies. They may not reflect the local conditions at your reach. So if you see these adult dragonflies or flying beetles or mayflies, they don't count towards this method. In a dry reach, you want to be careful about terrestrial or semi-terrestrial lookalikes of aquatic organisms. There are some insect larvae you can find in a dry stream bed that have a terrestrial origin but resemble aquatic insect larvae. Probably the best example are things like stratiomyids or soldier flies or other types of dipteran larvae. As a rule, these will have slightly more leathery skin when they're terrestrial versus aquatic, but that's not always the case. Snails, terrestrial snails are quite common in certain areas. They often have more robust shells than you find in aquatic snails, but that's not always the case. If the aquatic organisms you're observing are mosquito larvae, you don't want to count them. They're easy to recognize because they usually hang out at the surface of the water with a breathing tube that sticks up to the surface, and they have a very distinctive wriggle that when you disturb the water, they start sort of dancing around. Because they're so quick at colonizing standing water, that their presence isn't really indicative of the duration of that water. You can document their presence, but you don't count them in your assessment, in your tally. So I mentioned this earlier, both field and lab processing of invertebrate samples are acceptable for this method. When you're relying on your field identifications, you want to make sure that you have at least one crew member that is adequately trained. We recommend retaining voucher specimens where possible to confirm those identifications, and try to use hand lenses or field scopes. This is particularly important because some of the family level identifications are pretty difficult without seeing some of the mouth parts and smaller features, particularly if you're new to trying to do this. If you've got experience, you can probably get by with the unaided eye. Lab processing can be a better option if your field crews don't have the expertise. You can generally get a slightly more confidence in the identifications and abundance because you can, re-identify and recount the bugs that were collected. And this can save actually a lot of time in the field, so it can actually be cost effective. I would recommend that, of course, pick the approach that best suits the skills and capacity of your field crew. If you're doing field sorting and identification, you want to remove, tally, and identify every aquatic organism that you find. We recommend using white-backed sorting trays like the one pictured here. Featherweight forceps or eyedroppers, like there's an eyedropper photo in the photograph. These can be really helpful for removing them from the tray as you count them. You can put them in a petri dish like we show there. I recommend patience is good. Sometimes it might not look like there's anything, but you give the tray a little time to settle, and you'll see the bug starting to swim around. And again, we recommend that you collect specimens to confirm identifications later as possible. When you're counting up the number of individuals in a taxon, you basically count up until 10. After 10, you don't need to continue counting the abundance of that taxon. Now, the thing that probably is the most distinctive about the Western Mountains method as opposed to the Arid West method is the use of perennial indicator families. These are 17 families of aquatic invertebrates that the Xerces Society identified as indicators of perennial streamflow. And these are also used in the Pacific Northwest method. And I want to clarify that when they're known as perennial indicators, that means that they have a preference, that when they're found, you're typically in a perennial reach, but that doesn't mean they are exclusively going to be found in perennial reaches. They often occur in adjacent non-perennial reaches that are well connected to perennial reaches, as well as in intermittent reaches with particularly long durations. So again, their presence is not by itself diagnostic, but it is an important indicator for this method. Some of them are pretty easy to ID in the field, but others do require some training and practice, particularly because you need to learn to distinguish them from similar looking non-indicators. The user manual contains an appendix that shows every family and selected non-indicator lookalikes. Today, our goal is to provide you with an overview of these families, but it is not going to provide you with the training you need to actually identify and distinguish these families in the field. So you do need to get some more training on that specifically. I see a hand raised. Does somebody have a question? Or is that left over from?
No, hi, Rafi. This is Tracy. Just a quick heads up to folks. One of the things that the Xerces Societies did when they worked with us to develop these perennial and intermittent indicators, they developed an awesome field guide that we use for the SDAM in the Pacific Northwest. It is available on the Pacific Northwest SDAM regional webpage for download. It's truly a wonderful field guide. And so if you print that off and laminate it, it could be incredibly helpful in the field because, Xerces did a marvelous job. So that's just a quick heads up for a resource.
It is a great resource, and we actually have a link to that manual in the user manual. I talk a little bit about, let's talk about some resources. So these aren't, specific endorsements, but these are products that I've used and found very helpful. If you're trying to do lab identifications, there are some standard keys out there, and the two on the left are probably among the most widely used, globally for identifying families of aquatic invertebrates. There aren't tons of field guides out there, but the two that I'm showing here on the right, I think are actually quite good with really great illustrations and key diagnostic features. They tend to be, there are a number of other field guides that are aimed at fly fishers, and those are actually really good too. I just don't have them photographed here. But consider expanding your library with any of these books. So there are a number of really good online resources. There's this website that I've actually been using a lot lately called, it's an atlas of common freshwater macroinvertebrates of Eastern North America. And even though it's for the East, it works very well for Western taxa at the family level. And the website itself actually has a great interface, really high quality images with diagnostic features highlighted. I have actually made extensive use of some of their imagery in the material that we put together. It's specifically for insects, so if you're only interested in non-insects, the Colorado Division of Wildlife has an online field guide to freshwater mollusks. I apologize in advance if that link doesn't work. It seems to, they sometimes take it down and put it back up, but it is a great guide to the snail families that you're likely to observe in the Western Mountains. And the University of Minnesota has a really great program to help volunteers develop their own skills and for their stream monitoring programs. Now, I think it's there's a number of professional societies that can also provide workshops to help people gain expertise to identify these families. The ones I will highlight, so Tracy mentioned the Xerces Society. I think they would be, really excellent to provide these types of trainings. The Southwest Association of Freshwater Invertebrate Taxonomists covers, a good chunk of this region as well. Basically everything west of the Continental Divide and south of the, everything except for Montana and the Northwest. And then finally, the Society for Freshwater Science is a international organization, and they offer regular workshops. We're recommending that people contact regional coordinators if they want to organize a workshop, and they may be able to do it through these societies or other outfits.
I raised it up too.
All right, so let's start talking about these perennial indicator families, and we're going to start with the mollusks. So there's two major groups of mollusks out there. There's bivalves and snails, and they're pretty easy to recognize. And the Xerces Society identified the two families of freshwater mussels as perennial indicators: pearly mussels, Margaritiferidae and Nuionidae. They're very large, tend to have fairly asymmetrical shells. Many of their species are protected or endangered, and they can be extremely long-lived. So if you find them, they're really cool. And while the Xerces Society didn't identify this group, zebra and quagga mussels are something you also may observe, and they're based on their life histories, are also well adapted to perennial reaches. They're typically found in reservoirs and lakes rather than in rivers, but you will find them in rivers that receive effluent from an infested lake. Among the snails, Hydrobiidae is a particularly good indicator. These snails all have a right-handed opening. So if you're looking at the spire, the hole is on the right, and they have sort of these whorls with like a pretty good indentation. That's pretty distinctive. What I'm showing here is the New Zealand mud snail. You saw this photo earlier. Some authorities put it in a different family, whereas others treat it as part of Hydrobiidae. So either of these two families can count. There's no other species of Tatiidae in North America other than the New Zealand mud snail. Pluriceridae is another group. They have they're generally larger than hydrobiids. They have an operculum, which can be hard to see, like in this photo. They're a little bit less deeply whorled than the hydrobiids, but many of them are also protected species. And finally, freshwater limpets, pretty distinctive, hard to mix up with anything else. So if you see these, those are the other indicator family. And that's great, but yeah, you also have to know what else is out there so that you're not getting confused with them. So among bivalves, you have freshwater clams and fingernail and pea clams. They're pretty easy to distinguish based on their more symmetrical triangular shape. And among the snails, there's diverse families. One of the most common ones is Physidae, and they're really easy to distinguish because they're a left-handed group. So the hole is on the left when the spire is pointing up. Ram's horn snails are also pretty easy to distinguish. But then there's there's several families that might superficially resemble the Pleurocerid group, and you'll need to look closer to see if there's an operculum and how many whorls you have and things like that. I see Tracy and Ken are adding some additional resources. There's tons of really good ones out there, so I encourage you guys to share ones if you find one that you really like. So the next set of indicator families are the stonefly and caddisfly groups. There's two stonefly families. The common stoneflies are Perlidae, and the giant stoneflies are Pteronarciidae. These two groups are among the larger and more distinctive stoneflies, so they're pretty easy to identify. Although to to learn to recognize them, you really have to look at their mouth parts and their gills. This group always has distinctive gills that you could be able to see with some magnification, and they have some distinctive mouth parts that are kind of hard to see in the field. These giant stoneflies don't look like any other group of stoneflies, so they're pretty distinctive when you see them. They're very this isn't even the largest one. They get a bit larger than that. Among the caddisflies, there's 4 families that are perennial indicators, one of which is a case maker. It's called the saddle case maker, and it's unusual in that the case that it has 2 openings. There's a free-living group called Rhyacophylidae. They often have a greenish-blue tinge to their body, so they're pretty distinctive. And then you have the net spinner family. These hydrocychids build large nets that they retreat into and collect organisms and detritus that gets caught in that net and eat and consumes that for their food. And here's a nice photo of a caddisfly crawling into a retreat. Sometimes the retreats can be quite large. It can include some, almost entire cobbles. And finally, we have this family called fingernet caddisflies. They actually build a retreat like the hydrocychids, but they're more often found outside their net, like the rhyacophylids. They have these really funny long-shaped heads and some T-shaped mouth parts that are pretty distinctive. There are, of course, many other groups of stoneflies and caddisflies that aren't perennial indicators, as well as, I put mayflies here because mayflies are, pretty important to learn to recognize, and none of them are perennial indicator families. In general, like I said, the stoneflies, those 2 families are pretty distinctive. This one family, Perlotidae, I think is superficially actually quite similar to the perlids, but they don't have any gills that are obvious on the thorax, so they can be pretty easily distinguished that way. Caddisflies, there's tons of case maker species out there. Most of them, except for the glossosomatids, are not going to be perennial indicators. There are a number of other insect groups that contain perennial indicator families. We have 2 families of dragonflies and 1 family of damselflies. These ones are, you have to look at the shape of the mouth parts, whether they cover the face, they have these jaws that extend from the face, and if they're held in front of the face versus under the head, and also the shape of the antennae. You can see the jewel wings. They have these very long first segment of their antennae, so that's how you know this family. And beetles are a very diverse group, but 2 of the most distinctive beetles are the ones that are perennial indicators. The elmids, which are small and C-shaped. They always have the gills at the end of the abdomen that they keep inside like a lidded container. The adults are small with very large claws and have these they often have this stippling on the hind wings. So they're pretty distinctive and easy to distinguish from other families. And water pennies, these look like nothing else. They're flattened and round. This is a ventral view. You can see gills on the abdomen. They cling tightly to cobbles in fast-flowing water. And finally, we have the helgramites, which have these really big lateral extensions along the abdomen. They have 2 prolegs on the end of the abdomen, each of which has 2 claws. These guys are large and pretty hard to mistake for anything else. So with a little experience, you'll learn to recognize these as well. But like I said, every group has a lot of similar looking non-indicators. With the dragonflies and damselflies, I think that's actually a pretty tough challenge to gain this experience. The other groups, these beetles don't look anything like elmids or water pennies. They have much more, elongated, robust-looking larvae. The adults are pretty different too. They don't have that stippling and the large claws at the end of their toes. And this other group of related to the helgramites, these guys don't have sort of double claws at the end of their abdomen. There's a lot of aquatic insects in the dipteran group, the true flies. None of them are aquatic indicators, but they are really important for the overall abundance in the stream. Some of the more conspicuous ones include midges or Carnomidae, which are these sort of legless, C-shaped bodies, but with a distinct head capsule. Can vary widely in size. Some of them are bright red colored. Black flies have kind of like a bowling pin shaped body and these 2 fans at the end. They are only found in fast flowing water. And crane flies, when you find them, they have no distinctive head. Their head's actually retracted into their bodies. So these are sort of like the typical, maggot form. But these are large, robust-looking larvae that you can find quite often. And finally, the true bugs, hemipterans. These have wings similar to beetles, except that instead of being fully hardened, they're only hardened halfway, and they all have like a piercing mouth part. And again, none of them are perennial indicators. There are, of course, other aquatic invertebrates to look for when you're out in the field. Crustaceans like crayfish or amphipods or isopods, flatworms, segmented worms like this leech. Julie actually got this great photo of a bryozoan that she found in a creek near her house. And these sort of colonial organisms, these all count as aquatic invertebrates when you're trying to estimate the total abundance. You can find perennial indicators in a dry reach. So I've given you an example already of this helgramite in the Corydalidae family. Some of them can live in these chambers throughout a dry period in certain streams. So they're not going to be absent from intermittent reaches. You might find hydrocycid nets like you have right here in a dry reach. You might find exoskeletons as well. So if you find them, you should count them. You should count them as perennial indicators. When you record them on the field form, we have a reminder of what families count as perennial indicators. You write the name of every taxon, you estimate its abundance, or you tally its abundance up to 10, and then you check, is it a mayfly? Is it a perennial indicator? You can't be both. The rest will be something else. And then you want to calculate the metrics that are used in the actual SDAM: total abundance, abundance of mayflies, abundance of perennial indicator families, and number of perennial indicator families. Photos, again, are recommended if you're not collecting organisms. So that was a lot. I think this represents probably one of the more important but perhaps more challenging indicators to measure for this SDAM. Are there questions about it based on what we've discussed today? I know Tracy had mentioned that in the Pacific Northwest that there was some challenges in getting people to accept a method that included this method, and that's why she invested in developing that identification guide from the Xerces Society. So make sure that you have those resources available to you if you encounter people who have concerns about measuring aquatic invertebrates.